Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET question paper for the subject Botany conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CUET various other entrance exams, you can go to my channel Omni Bio Entrance. In the channel playlist C for CUET exams, there you will find. Coming to the first question, the causal agent of byssinosis is. So byssinosis, it is also known as brown lung disease or mill fever, and it is caused due to inhalation of cotton dust. Particularly it is seen in those working in textile mills, cotton processing facilities, etc. So the answer here is option 3, that is cotton dust. The next one, the importance of characteristic of case selected population is convex survivorship curve. That is a type 1 survivorship curve, so that is correct. A low fecundity, yes, the fewer offsprings are produced, so that is also correct. Exist close to their carrying capacity, that is also correct. Intraspecific competition, yes, that is competition among individuals of the same species for various resources like that. So that is also seen. An example of case selected species is human beings. So the answer here is option 3, that is A, B, C and D. Then next one. Match the following process with the changes. Nudation is a creation of barren area. So A1 is seen in option 1 and option 2. Okay, we will see B. Exesis is establishment of species. So B2, so that gives you the first option as the correct answer out of 1 and 2. Then reaction is modification of habitat, and stabilization is community in equilibrium with the organism, D3. So A1, B2, C4 and D3 is the answer. Then given below are two statements. One is labeled assertion, other is reason. So assertion. The zone of transition between two communities is called an ecotone. That's correct. The ecotone has more diversity than either of the two communities. Okay, if there is an zone of transition between two communities, the ecotone will have more species diversity than either of the two communities. Okay. So both these statements, both A and R are true, but R is not the exact reason for A. Okay, why it is called ecotone? There is nothing like that even though, but that is not the reason. So that doesn't explain why it is called an ecotone, that it has more diversity than other two communities. It is the assertion and the reason are just like any other two different statements. So A and R are true, but R is not the correct explanation of A. Then next one. The decreasing order of concentration of greenhouse gases in the atmosphere. So the one with highest concentration is carbon dioxide out of this option, and the lowest concentration is for N2O. So after carbon dioxide, the next highest is methane, CH4, and then comes CFCs. So carbon dioxide in the beginning, N2O at the end, CH4 second and CFCs the third. So your answer comes to be A, B, C, D, that is 1. Then next one. Inverted ovules are called as. So in inverted ovule, The micropyle will be facing the base of the ovary. Okay, it is also called anatropous. See the diagram here, that is this one. So it is inverted ovule. So it is second. So the answer here is option 2. Now orthotropous means it is an upright ovule, as you can see here, with the micropyle facing the apex of the ovary. And amphitropous means the ovule with this one. Ovule with curved or bent shape. Okay, this is it. That is amphitropous. And yes, that is amphitropous. And hemianatropous is this one, which is partially inverted orientation. That is intermediate between anatropous and orthotropous. That is hemianatropous. So these are the various types of ovule. I have explained only those which are given in the options. Then the different methods to overcome self-incompatibility are mixed pollination. Yes, that is a method to overcome self-incompatibility. Okay, before that, what is self-incompatibility? It is a mechanism to prevent self-fertilization in plants. So self-incompatibility means it favors cross-pollination. Here they have asked what are the methods to overcome this self-incompatibility. So how will you overcome self-pollination? So mixed pollination, it is the method where the stigma is disguised from recognizing the incompatible pollen. Okay. So here the stigma is pollinated with a mixture of live incompatible pollen and killed compatible pollen. So the stigma will recognize only the live incompatible pollen and it will help in the growth of the pollen tube. So mixed pollination is correct. Then you have bud pollination. So in bud pollination, so pollen collected from the anther of one flower is applied to the stigma of another flower on the same plant. But the flower is not fully opened. That is bud pollination. Stub pollination, it is also somewhat similar to bud pollination. But here the pollen is applied to the stigma after the flower's anthers have been removed. That is stub pollination. Then intra-ovarian pollination is the case where you directly inject the pollen into the ovary of the flower. So all these mechanisms, that is A, B, C and D, they are different methods to overcome self-incompatibility. That is, they will favor the self-pollination mechanism. So answer is option 3, A, B, C and D. Then match the list 1 with list 2. So the process and the scientists who have discovered them. So intra-ovarian pollination, that was discovered by Kanta. A4. A4 is there only in one option, so that is going to be your answer. All the others have A as 1. That is just to confuse you. Okay. Okay. A4 is the answer. It is Kanta. Double fertilization, it is by Navastin. So B is 3. Singami by Strasberg. So C comes 2. Yes, C2. And necrohormone theory is by Haberland. So D 1. So answer is option 4. Then next one. Okay, assertion, reason, question. So assertion. Male sterility in angiosperms can be induced by inserting a bacterial gene called Barnase. Yes, that is correct. Barnase is isolated from bacteria and it is introduced into the angiosperm to induce male sterility. Then Barnase causes premature breakdown of tapetum resulting in arrest of microspore development. So tapetum is involved in microspore development. So the Barnase genes, once you introduce into the angiosperm, it will cause breakdown of the tapetum, and your microspore, that is your pollen grains, they are not going to develop. So this reason it explains the assertion. So both A and R are true, and R is the correct explanation of A. Then next one. The most effective sugar in promoting pollen germination, it is sucrose. And that's a direct question. So answer is option 3. Then next one. The specimen collected from the same locality from where the holotype was collected is called topotype. Isotype, it is a duplicate specimen of the holotype and it is collected from the same locality. Okay, it is a duplicate of the holotype. It is not the specimen collected from the same locality from where holotype was collected. It is topotype. Then syntype is one of two or more specimen used to describe a new species or taxon within when no single holotype was designated, that is called as syntype. And paratype, it is used to supplement the holotype, that is it provides additional information regarding the holotype, that is paratype. So your answer here is topotype. Then next one, free central placentation is seen in the family Caryophyllaceae. Answer is option 3. Brassicaceae, it exhibit parietal placentation. Cucurbitaceae, it exhibit parietal or basal placentation. Capariaceae, it is parietal placentation. So the answer here is free central placentation, it is by Caryophyllaceae. Then next one, choose the correct answer from the options. They have given taxonomic characters and their family. So jaculators, they are present in which family? So jaculators, they are present in the family Acanthaceae, so A4. A4 is there only in one option, so 1 must be your answer. Caryopsis fruit is seen in Poaceae, B3. Cyathium, it is a type of inflorescence seen in Euphorbiaceae. Cremocarp fruit, it is found in Apiaceae. So A4, B3, C2 and D1. Answer is option 1. Then next one, assertion reason. APG classification system is not useful for adoption in herbaria and flora. Yes, that is correct. APG system taxa have been classified only up to the level of family. That is the reason why it is not useful for adoption in herbaria and flora. APG classification system. So both A and R are true, and R is the correct explanation of A. Then important character of family Asteraceae is. So Asteraceae is the family where you can see your sunflowers. Okay, we will see. Inflorescence is racemose, head or capitulum, that is correct. Presence of Cypsella fruit, yes, that is the characteristic fruit of Asteraceae family. Presence of Gynecostegium, it is found in the family Apocynaceae, not Asteraceae. And placentation, it is in Asteraceae, it is basal or axial placentation and not free central. So A and B are the only correct answers, so 1 is the answer. Then Hanstein proposed. So Hanstein proposed the histogen theory. That is option 2 is the answer. Then given below are two statements. One is labeled assertion and the other as reason. Assertion. Root apical theory of Nagelli does not hold good for flowering plants. Yes, he proposed that theory for the non-flowering ones. So he proposed that the root apex is organized around a single apical cell. But flowering plants, they have shown that the root apex is actually organized a group of initial cells, not a single apical cell. So root apical theory, it does not hold good for flowering plants, and the reason they have given is in flowering plants a group of initials have been observed. Yes, so that is why the root apical theory doesn't hold good for flowering plants. So A and R are true, and R is the correct explanation of A. Then next one. So the type of stomata and the family to which it belongs. So anamocytic stomata, it is found in Papaveraceae A4, that is there in option 4 only. So that must be your answer. Then anisocytic stomata, it is found in Apiaceae. Parasitic stomata is found in Magnolinaceae, and diacytic stomata is found in Acanthaceae. So A4, B3, C2 and D1 is the answer. Then the anomalous distribution of vascular tissue in older stem of Bougainvillea is due to. So anomalous distribution of vascular tissues in Bougainvillea, it is due to formation of accessory cambial rings. Formation of interciliary phloem, no. Abnormal behavior of normal cambium, abnormal behavior of abnormal cambium, all that are not correct. It is due to formation of accessory cambial rings only. So A only is the right answer. Then Malacophilus leaves are found in. So Malacophilus leaves means they are soft leaved and describe the plant which is fleshy and succulent, and it can store large amount of water. So naturally such characteristics they are found in the case of xerophytes, which are growing in dry habitats. So A is the right answer. No, sorry, 2 is the right answer. Then which amino acid contain the amino groups NH2 attached as a side chain? So they have the amino group in their side chain, not the alpha amino group. Alpha amino group and alpha carboxyl group, they are characteristic in structure of all kinds of amino acids. But here the side chain group or the R group is having an amino group. So that is seen in the case of lysine and arginine. Answer is option 4. So this question means you should be thorough with the structure of various amino acids. All the 20 amino acids you have to be perfect. So lysine and arginine is the answer. Then the ideal value for the backbone dihedral angle of antiparallel beta sheet. So as per the Ramachandran plot, you have got the phi and the psi angles. They have asked which of these is the one for antiparallel beta sheets. So for beta sheets it is phi 139 and psi 135. So B only is the answer, whereas others are not the dihedral angles for antiparallel beta sheets. So 2 only. Then next one. Then they have given chemical nature of the side chain of various amino acids they have given, and they have asked for the amino acid. Again you should be thorough with the structure of various amino acids as well as their classification, such as aliphatic amino acids, aromatic amino acids, basic amino acids, acidic amino acids. All these you have to be thorough. Also hydrophobic, hydrophilic amino acids also you should know. Okay. Now aliphatic amino acid, that is your out of these it is valine. So A3 is there only in option 4. They have purposely given A as 1 in all the 3 options so that you will get confused. So aliphatic amino acid is there only in valine, A3. Sulfur containing amino acid is present in cysteine, that is B4. Then imino group in the amino acid is present in the proline, and amide group is present in glutamine. So A3, B4, C1 and D2 is the answer. Then next one. Myoglobin is a globular protein which is made up of how many amino acids? It is 153 amino acids. Answer is option 2. Then which types of protein is commonly found in RBC? So RBC is having both spectrin and ankyrin proteins. So answer is option 4. These are just direct questions. Then N-acetyl glucosamine and N-acetyl muramic acid in the cell wall of bacteria are connected by. So they are connected by beta-1,4 glycosidic bonds. Answer is option 1. Again a direct question. Then what is the basic property of gram-positive bacteria? So gram-positive bacteria, you all know they have a thick peptidoglycan layer. That is the characteristic feature of gram-positive bacteria, whereas gram-negative bacteria, they have thin layer of peptidoglycan. So thick peptidoglycan layer is present, that is correct. Echoic acid are present, that is also correct. Periplasmic space is negligible, that is also correct. Outer membrane is present. Outer membrane is present in the case of gram-negative bacteria, not gram-positive. So A, B, C only is the answer. Answer is option 2. Then again match the following. So halophile. Halophile, happily you can go for halobacterium. They grow in saline conditions. So A will be 3. A3 is there only in one option. So 4 is the answer. Acidophile. Acidophile is your sulfolobus. Thermophile, that is Thermus aquaticus, C1, and microaerophile is your Treponema. So A3, B4, C1 and D2 is the answer. Then agrobacterium rhizogenes, not tumefacients. They have asked regarding rhizogenes. So rhizogenes, they have Ri plasmid instead of the Ti plasmid, and they cause hairy root in plants. Is used to genetically engineer the plant because of the following statement. So which of these statement? Why agrobacterium rhizogenes is used in genetic engineering? Because they produce hairy roots, which are roots that grow abundantly and rapidly, and these hairy roots they have various characteristics like high biomass production, and they produce secondary metabolites also. So it contains a Ti plasmid. No, it is Ri plasmid in agrobacterium rhizogenus. Ti plasmid is found in agrobacterium tumefacients, not rhizogenes. Then it causes gene transfer to plant. Yes, it does cause gene transfer to plant, but that is not the exact reason why it is used to genetically engineer plants. It infects all type of plants. Yes, not all type of plant, but a wide range of plants it infects. Okay, so that's also not correct. Infect plants and produce a good root system. Yes, that is why agrobacterium rhizogenes is used to genetically engineer the plant. Then next one. Which of the following technique is used to inactivate gene by altering the DNA? Homologous recombination. Yes, that is used to inactivate genes by altering the DNA or altering the DNA sequence. Okay. Now how is it done means you have to identify the target gene which has to be inactivated. Then you create a DNA construct with a modified version of the target gene. Then you introduce this DNA construct into the cell, and this DNA construct which you have introduced in the cell will replace the target gene by this modified version of the DNA. So here there is alteration in the DNA sequence. So it is homologous recombination. It is a technique used to inactivate a gene by altering the DNA. Then antisense nucleic acid block. It is a technique that involves antisense oligonucleotide to block the gene expression. So it does not alter DNA sequence. So that's not the answer. Antibody, they are used to detect specific proteins, not to alter DNA sequence. Then introduction of dominant inhibitory mutant. Um, so you introduce a dominant inhibitory mutant or a dominant negative mutant, and this will inhibit the function of the gene, but it does not alter the DNA. So 4 is not the answer. So your answer is homologous recombination. Now, which is not a transmembrane protein? So out of these, it is ferredoxin. It is a water soluble protein. It is not a transmembrane protein, and it is involved in electron transfer reaction in photosynthesis. LHC is your light harvesting complex. Okay. So all the others are transmembrane protein. Then B gene of ABCD flower model is made silenced in a plant. Which of the word is absent in the sepal? So this is the ABC model. Here A, it codes for the sepals or the calyx, that is this region of the plant of the flower. Then A along with B, it will code for petals, that is the petals or the corolla. Then B along with C, it will code for stamens, and C alone, it will code for the carpels. So B, as you can see in the diagram here, it is involved in the development of stamens as well as petals. So once you silence the B gene in a plant, both stamen and petals will not be formed. So the answer here is C and D. Answer is option 4.